Hello everyone. I welcome you all once again to my series of lecture that is Understanding Pharmaceutical Science with Dr. A.G. Hariharan. So today we are going to discuss about the isolation of pure culture. It is one of the very important technique in the pharmaceutical microbiology as well as in microbiology as a whole. Because the important is that since the microorganisms are present in nature as a mixed culture, it should be isolated into a pure form. So this lecture I have elaborately discussed about what is meant by a mixed culture, pure culture, and what is the basic principle behind this isolation of the pure culture. And the second part of my lecture has been elaborately discussed about the various methods which can be used for the isolation of the pure culture. So let us understand this technique by basically understanding about a mixed culture. So if you see in nature, so if you take a small sample of your water or you take a small sample of your air or your soil, you can see that each sample carries a mixture of microorganisms. The bacteria, fungi, or different types of bacteria will be growing together in this natural environment. So it is little bit complex, and by it is very difficult for us to study a single type of microorganism which is present in this mixed culture state. So there is a need to isolate each and every microorganism individually from this mixed culture so it can be studied and it can be explored in a better manner. So there the need of the pure culture came into an existence. So if you see the development of pure culture, so the development of pure culture was done by the German bacteriologist who is the father of the medical microbiology is Robert Koch. So Robert Koch has put form his postulates in the medical microbiology in terms of Koch postulates, in that he states that a particular disease infection is caused by a particular type of microorganism only. So to state that this particular microorganism can cause this particular disease means he has developed a technique of isolating this pure culture of microorganism from the mixed culture. So Robert Koch has developed this pure culture technique, right? So now the concept arises. What is a pure culture? So the pure culture is nothing but a growth of population of cells generally arising from a single cell. So from a single cell, it divides on its own to form a colony. That is considered as a pure culture. So if an organism is present in this pure culture, only we can able to characterize it. Whether we can characterize its morphological characteristics like its cell wall characteristics or flagellar characteristics like this, or biochemical characteristics or genetic characteristics can be possible only in pure culture form. So how we can isolate a microorganism in pure culture? What is the basic principle behind it? If we see that, so we get a mixed culture as a sample, then it is diluted to such an extent that this will have relatively a low density of the population. Then it is evenly spread out on an agar surface. So what happens is this mixed microorganism will forms in an individual cell into the agar surface. Then it grows into an individual colony. So each cell divides and divides, then finally it forms a colony. So this colony we can see it macroscopically, then it is subcultured into a pure culture form of a microorganism. So by technically as per the definition, this principle will help to isolate a organism in pure culture form since it is arising from a single colony once it is evenly spread on a agar surface. So let us discuss what are the different methods we can use to isolate this into a pure culture form. So different methods of isolation of pure culture, if you see there are many methods are available. Out of this, three common methods are commonly used in the isolation technique. One is the pour plate technique, second is the spread plate technique, and third is the streak plate technique. So let us discuss each technique elaborately. The first one is the pour plate technique. So the pour plate technique is a very simple technique. If you see what actually we are doing is First, we will take a sample, then we will dilute serially until the microbial population is drastically reduced. Then it is added to an agar media which is in liquid state. Then it is poured into a plate so that it fixed its colonies at individual level. Then it grows into a pure culture form. So out of this, the major step is that getting at an appropriate microbial dilution. So if you see in this picture, so the original sample has been serially diluted to 10 to the power minus 1, minus 2, minus 3 and minus 4. We can get sample and we can make a pour plate at any stage. For example, I am taking a 10 to the power minus 3 dilution. I will get more colonies. So that is this much populated that we cannot able to isolate into subculture into a separate pure culture form. It will get sometimes overlapping. So get a particular dilution so I can see an individual colonies so that it can be easily isolated or subculture and developed or kept into a pure state. So how we can perform a pour plate technique is simple one. So in the first step, we have to perform a serial dilution of the sample. It's a simple technique. Take the original sample, take 1 ml of the original sample, transfer to a sterile test tube carrying a sterile distilled water around 9 ml, right? Then from this 1 ml, take it and dilute it to another 9 ml of distilled water. Perform this procedure until you will get a sufficient dilution, right? So 1 ml 9 ml, so we will get a tenth fold dilution, tenfold serial dilution we will get. If you take 5 ml, then it is a 5-fold serial dilution you can get it. So 10 to the power minus 1, minus 2, minus 3 and minus 4 dilution until you will perform, until the colony is sufficiently reduced so it can get into an individual cells. So second step is mixing in the liquid agar. So prepare a liquid, prepare a agar media, sterilize it in a conical flask. So to this, once the agar reaches to a temperature around 45 degrees Celsius, this is the critical part of this pour plate technique. So at 45 degrees Celsius, the agar will be in liquid state. If it is go below 45 degrees Celsius, it starts getting solidifying. So at this stage only we have to add a small volume of this diluted sample which carries the microorganism. So this temperature 45 degree will be sufficiently enough for the microorganism, especially bacteria or fungi, which is not killed by this brief exposure. So once it gets solidified, it can get fixed at a particular places as an individual cells. So we will add it once the temperature of the media is around 45 degrees Celsius, then we will mix it so it can equally distributed in the sterile agar medium. Then the step 3 is the pour plate. Then it is poured into an aseptic manner in a laminar flow hood into a sterile culture disc. In a petri disk, you just pour it until it forms a layer and allow it to solidify. Once it gets solidified, what happens is that the individual cell gets entrapped or fixed in a particular places of the agar. Then it is incubated. And the fourth step is the incubation. Then it is incubated in an inverted position at a particular temperature for a particular duration of time. So after that, you can see that each cell divides and it forms a colony. So that is considered as a pure culture. Then in the step 5, we will subculture it into a fresh medium. Then we will use it for our further study, either for a characterization work or some production activities, right? So this is the first technique, that is the pour plate technique. And the second technique is the spread plate technique. So when compared to pour plate technique, if you see, the spread plate technique is the most easiest way of isolation of pure culture, and it is also a direct way. It is a simple method. What actually we are doing is we will dilute the sample similar to the pour plate technique to a certain extent. Once the population has been reduced up to 300 cells, then a single drop is placed at the center of the petri dish. Then it is evenly spreaded. So that's why we call it a spread plate technique. It is evenly spreaded. It forms a layer. So after that, after incubation, we can see an individual cell will grow into a colony. So the spread plate technique is the most widely used technique for the isolation of pure culture. So if you see the stepwise manner how it can be performed is the first step. Similar to pour plate technique, we have to perform a serial dilution process. Take the sample, 1 ml of the sample is added to 9 ml of distilled water. So sterile distilled water. So we get 1/10 dilution. Then from this 1 ml transfer to another 9 ml of sterile distilled water, we will get 100 dilution of the original sample. So it is followed until we get a sufficient diluted sample so that it can be easily spreaded and forms an individual colonies, right? So the second step, what we are doing is we are preparing a media, we are making a plate of this media, then we will pipette a small volume into the center of the agar media aseptically, right? Then this is spreaded with the help of a bended glass rod. So first the bent glass rod is dipped into an alcoholic solution, then it is shown into the flame for the sterilization purpose, then it is placed over the drop. So with the length of the glass rod it gets spread into a single layer. The once it is rotated, what happen is that it is spreaded to the entire surface. So after that it is kept in the same position for 1 hour so that the each cell gets entrapped into that media, agar media. Then it is inverted and incubated at an appropriate temperature and duration. So during this time, each individual cells will start developing into an isolated colonies. Then finally, this colonies is get subcultured on a fresh medium to get the pure cultures. So this is the second and the most easiest method of isolation of pure culture. So apart from this isolation technique, the spread plate technique is used for other purpose, especially for the microbial counting, enumeration of the bacteria. The spread plate techniques are used. Otherwise, for the identification of the viable cells, CFU counting, colony forming unit countings, this spread plate technique is used. That is to calculate the number of viable cells are there. That is number of living cells are there. So number of living cells will generally number of colonies. So based on this, we can identify. And the third technique is the streak plate technique. So the name itself tells the streak means we are streaking the culture. So the first step of this streak plate technique is transfer of this sample mixed cell culture to a edge of an agar plate. So before starting this, what has been done is the petri dish has been divided into 4 sectors. So you take the petri dish, first draw a horizontal line, then draw a vertical line at the center, we will get 4 sectors. So this mixed culture is added to the first sector at a particular location. So we will get the culture with the help of an inoculating loop or a swab, you add a small amount of the culture, right? So the second one is streaking in the first sector. So with the help of a sterilized inoculating loop, so the inoculating loop is sterilized by showing into the outer blue flame of the Bunsen burner. It gets red hot, then allow it to cool for 10 to 15 seconds. Then you touch the mixed culture and make a streak, right? So this is called streaking of the first sector. So with the help of sterile inoculating loop, we will streak it. So we will get this one. So then we will rotate the petri disk. Then we will streak the second sector. So for streaking the second sector, the inoculating loops should be sterilized once again by showing into the blue flame of the burner and allowing it to cool after streaking the first sector. So then we will get the sample by simply touching the end of the first sector, then streak it into the second sector. This you can see from this picture, the second sector has been streaked, right? Then the next step is streaking of the third sector. So once again the inoculating loop is sterilized, then the sample is got from the second sector, then it is streaked in the third sector as shown in the figure. You can see it, it is streaked once again. Then finally streaking in the final sector, that is the fourth sector. So in this time, what we are doing is we are not sterilizing it, we are continuing the streaking in the fourth sector. So during this, what happened is this process of streaking from first to second, third, and fourth sector, the organism started getting diluted. So we will get an individual cells getting streaked in the final sector. So after incubation, this will develop into a separate colonies, right? This you can see from the picture how the fourth sector is streaked. Then finally, this has been incubated and it will get an individual colonies. Let us, I will show the sequence of steps. In the first step, you add a small quantity of the sample at an edge of the Petri disk. Then with the help of a sterile inoculating loop, streak to perform the first streak, then rotate the Petri disk, sterilize the inoculating loop, then do the second streak at 90-degree angle more or less. Then once again sterilize the inoculating loop, take the sample from second streak, perform the third streak. Then after that, perform the fourth streak. Then incubate it at an appropriate temperature and duration. We will see the colonies as an individualized one in the final streak. Then isolate it and grow it into a fresh medium. So this is the method how we will perform the streak plate technique. So thank you very much. I think this lecture will be helpful for you. You isolating the pure culture in our laboratory scale, or even for a production of some antibiotics or vitamins in an industrial microbiology. The first step you have to isolate in the individual colonies. So thank you very much. Thank you once again.